Welcome. Good morning, good afternoon, and good evening to those joining us for the first webinar in a series of webinars around the theme of engineered gene drives, the state of the research. This webinar series is brought to you by the GeneConvene Global Collaborative. My name's Dave Obrachta. I'm a member of the GeneConvene Global Collaborative team, and I'll be hosting and moderating this series of webinars focused on some of the technical aspects of their development. Before I get to today's speaker, I'd like to say a few words about the GeneConvene Global Collaborative, this webinar series and upcoming webinar series, and a bit about how we'll conduct today's seminar. The GeneConvene Global Collaborative is an initiative within the Foundation for the National Institutes of Health, and it's evolved from the FNIH's long-time involvement in the vector-borne disease space. GeneConvene is working to advance the safe, ethical, and rigorous exploration of gene drive and other genetic biocontrol technologies by anticipating emerging issues and building consensus for best practice and guidance. GeneConvene offers accurate information, advice, and technical support for the purposes of fostering responsible approaches to research and governance of gene drive and other genetic biocontrol technologies for public health. Let me also add that GeneConvene thinks that the responsible exploration of genetic biocontrol technologies is critical because the potential of these tools to contribute to malaria's control and elimination, as well as other urgent public health challenges, is enormous. I encourage you to learn more about the GeneConvene Global Collaborative by visiting the FNIH website, fnih.org, and just searching for the keyword GeneConvene. Let me also point you to the GeneConvene Virtual Institute. This is a knowledge base that aggregates, tracks, and shares the latest about gene drive, from scholarship and research to media reports to policy and regulatory documents. This is part of an effort to create a space where those interested in gene drive can come and see what is happening and what has been happening. The Virtual Institute publishes a weekly newsletter that will help you keep abreast of activity in the gene drive space. You may not be aware of it, but since January of this year, there have been at least 120 scholarly publications in the gene drive and genetic biocontrol spaces. And there's been at least 170 media reports. So one of the reasons the Virtual Institute was created was to help you keep up with all of that activity. There are important ongoing discussions and conversations concerning these technologies and their potential applications. The Virtual Institute can assist those involved to become informed and to stay informed. So I also encourage you to follow us on Twitter and Facebook. Let me say a few words about this webinar series, the ones that'll follow. This particular series of 6 seminars is focused on the science and is intended to provide researchers an opportunity to share their latest results and to receive feedback and discussion from others. While this series is limited to 6 researchers in an ever-growing field of research, we'll be coming back to this technical theme in later webinar series, and we'll have an opportunity to hear from others who are engaged in this type of research. Following this series will be a series that explores the themes of regulation and policy. And after the new year, we'll continue with other themes, and you should stay connected with us to keep abreast of what those offerings will be. Let me just give you a few words of orientation here for today's seminar before I introduce today's speaker. If anybody has any technical issues during the course of this seminar, you can contact our FNIH events staff, who will be monitoring this webinar, this email address, events@fnih.org. The presentation is scheduled to be roughly about 50 minutes. Your video and audios will be muted. We'll take questions after the presentation, and we'll do that using the Zoom chat function. The questions will only come from identified questioners, and the questioner's identity won't be made public, just for privacy issues. We have 90 minutes to engage, and we'll end at the end of 90 minutes, and any remaining questions will be addressed offline. The presentation itself will be recorded, and it'll be posted on the GeneConvene Virtual Institute site at this URL shown below. Okay, let's move on to today's speaker. It's my pleasure now really to introduce today's speaker, Dr. Anthony James from the University of California at Irvine. Tony's the Donald Bren Professor in both the Molecular Biology and Biochemistry Department in the School of Biological Sciences, as well as the Microbiology and Molecular Genetics Department in the School of Medicine. Tony's a member of the National Academy of Sciences. Tony's had a long involvement in the field of vector biology, beginning as a postdoc at Harvard many years ago, where he investigated the salivary gland secretions of mosquitoes, and was actually the first to clone one of the many genes responsible for making the important cocktail of proteins found in mosquito saliva. He's also played important roles in developing transgenic technologies for mosquitoes, and was the first to create a transgenic mosquito using what are now widely available technologies. He's had a longstanding interest in the development of genetic biocontrol technologies for mosquitoes, and has been developing homing type gene drive systems that he hopes to couple with anti-parasite effector genes that will enable the modification of standing population of malaria vectors. And I'm sure we're going to hear lots about that today. He's the principal investigator of the UCI Malaria Initiative. It's a research consortium focused on gene drive technology development with the goal of malaria eradication. So, Tony, good morning to you in California. It's a great pleasure to have you with us. Thanks so much for coming. The floor is yours just as soon as we can transfer the controls over to you.
Great, David, thank you. And while we're waiting for the controls to be transferred here, I want to thank David for.
Okay, I think you can go ahead and share. Mm-hmm.
Okay. Well, not yet. It says only the host can do it. Let's try it again. No. I'm still getting a host only right now message.
Okay. Tara, can you look into that?
There we go.
Thanks.
There we go. We're set.
Very good.
Can you see it? David, can you see it?
Good. Yes,
okay,
sir.
Great. Thanks. Well, I want to thank David and Dietrich and Tara for helping me get this sorted out here and allow me to be the inaugural speaker in this series here. And as David indicated, I'm going to be talking about gene drive systems for mosquito population modification. There are a number of institutions that are involved in this work. You can see them across the top here. And there are key researchers in each of these that have, whose data I'm going to be showing today. But we won't have a chance to shout out to everybody, but I hope I've included everyone here. And we enjoy funding from the University of California Malaria Initiative, and past funders are Bill and Melinda Gates Foundation, NIH, Keck Foundation, and Tata Institute for Genetics and Society. And as David said, we are part of a consortium called the University of California Malaria Initiative, which comprises 4 campuses at the from the University of California, along with Johns Hopkins School of Medicine. The mission of the UCIMI is to promote the discovery and development of novel science for the goal of malaria eradication. And for that, we are pursuing genetic strategies for controlling the transmission of malaria parasites. So the objectives of genetic control are twofold. One is population suppression, and you can consider this a genetic analog to insecticide, and basically the idea here is to reduce or eliminate mosquitoes. But an alternate approach, which we're employing, is called population modification. It's also been called population replacement historically, and also alteration. But we're talking about changing the mosquito's ability to transmit pathogens. And we're doing this by essentially a synthetic approach, by engineering genes that confer resistance to malaria parasites and moving them into vector populations. So population modification, once again, is to engineer mosquitoes with genes to prevent malaria parasite transmission. It's based historically on the genetics of vector competence, where it was shown that individual genes could have a very large impact on whether or not a specific vector could transmit a specific pathogen. And the hypothesis of the research is that these genes could somehow be introduced into mosquito populations. They could result in this particular case in less malaria. So these are genes that are conferring resistance to malaria. They should result in less disease and death. So there are epidemiological endpoints to this research. So why population modification? Well, we think that it can have a very sustainable impact on malaria elimination during all the stages of malaria, the lead up to malaria eradication, control, pre-elimination, elimination, prevention, and reintroduction phases. It doesn't leave an empty ecological niche. That is, the mosquitoes are still there. They just cannot transmit malaria. And the modeling has shown that it's likely to be stable in low vector population densities. So this allows a serial approach to resource control, meaning that one can go from one area to the next area, attempting to get local elimination of malaria, and ultimately to a consolidation of gains where that'll subsequently contribute to eradication. The targets that we're going to be talking about today are two vector species, Anopheles gambiae, which is in sub-Saharan Africa, Anopheles stevensii, which is in southern Asia, but now has been apparently invading the southern horn of Africa. And our primary target is the human malaria parasite Plasmodium falciparum, which causes the majority of morbidity and mortality related to malaria. So we, a long time ago, talked about three major research areas. The first was the molecular genetic manipulation of mosquitoes. This is something that was done in a laboratory and basically was trying to meet the objective of making mosquitoes that carry anti-parasite genes. And there was a tremendous amount of work that went into this, not just from our group, but lots of groups. And David alluded to it. This involved identifying genes that could control the expression of effector molecules, identifying the effector molecules, and developing transgenesis that allowed those effector molecules to be put into mosquitoes. Once one had such a strain, it was necessary to ask, okay, is it possible to come up with ways to move these developed genes into wild populations? And these wild populations, or wild type, if they're in the laboratory, are ones that did not have the gene. And the idea was, can we come up with a way to move them quickly into that? Then, of course, we need to know a lot about the field, all right? I have pictures here of a village in sub-Saharan Africa, a village on the banks of the Amazon River, and an urban area in southern Asia. And you can tell that these are very different ecosystems. However, they all have one thing in common, which is that there is malaria transmission in these, parasite transmission in these areas. And so it's important to know what you're talking about when you talk about the field. And you can see it can be very different here. So assuming that you've made such genes and put them into mosquitoes, and we've done that, how can these actually be used? Well, it's possible to do what are called inundated releases of mosquitoes. And here we're talking about carrying an anti-parasite gene, which is in green. And these are mosquitoes here that have parasite-resistant genes in them. And what happens is one releases these into the wild or in the presence of wild-type mosquitoes. They mate with the wild types, and eventually you start to get, with subsequent releases, introduction of this gene into the population. And you need to do this enough so that all the mosquitoes end up being resistant, or a large fraction of them do. Another approach is one where you combine the anti-parasite genes with something that carries a dominant, or with a construct that's a dominant lethal. And here the idea is that when you release them, at every generation that you lose a certain proportion of the population, which is the dominant lethal in red. And ultimately, you suppress the wild population while at the same time increasing the frequency of the ones carrying the beneficial traits here, the anti-parasite genes. The third thing is something which is called gene drive, which is what we're going to spend all the time talking about this morning, where you link the anti-parasite to some mechanism where when you have a single release, the release of these insects interacting with the wild types will lead to all the subsequent mosquitoes carrying the genes of interest, the beneficial genes. So what is gene drive? Simply spoken, it's a preferential inheritance of a genotype. And this is usually seen in diploid organisms during sexual reproduction. It's been called a violation of Mendelian patterns of inheritance. I prefer circumvention. It is basically going at the aspect of random segregation that came out of Mendel's work. And we talk about a drive mechanism, which is the underlying biological feature that's incorporated in the system that causes this drive. And the GeneConvene series is going to be featuring a number of these mechanisms, okay? And we're going to be talking about one in particular today, but these mechanisms then form the basis for developing a system, which is a final synthetic product that achieves this preferential inheritance. So the mechanism that we're going to be talking about today is one that is currently talked quite a bit about in public and scientific literature, which is the Cas9 guide RNA mediated gene drive. So this is just a simple representation here of the genetics of gene drive to further emphasize this point. If we have standard Mendelian inheritance where we have an organism that is homozygous for a gene that we're interested in here, this is a parasite resistance gene, crossed with a wild type, in this case, one that is susceptible, we would expect under these circumstances a homozygote resistant cross with a homozygous susceptible to get a heterozygote. We do a test cross by back crossing it to the susceptible, and we would expect 50-50 segregation. With a gene drive circumstance, we form the same type of cross here. We get a heterozygote, do the test cross, and we get 100%. So this is a very straightforward assay for gene drive, is to do this type of crossing, to do this back cross and look at what the progeny are. And we're going to see a number of experiments in the talk here that feature this type of approach. So how do you make an autonomous gene drive system? So when we talk about autonomous, we mean something that continues to propagate once it's integrated into the mosquito. And we start here with a chromosome that has a target locus. And so this is where we would like to have our gene drive system integrate into. And then we build a plasmid. And the plasmid, it has encoded on it the Cas9 nuclease, which is an endonuclease. It also has guide RNAs, which interact with the nuclease that determine a specific spot in the target locus where we're going to have cleavage. And then we have cleavage by the endonuclease. And then we have homology arms, which allow interactions between our plasmid and the target chromosome based on complementarity of the DNA sequence. And generally, the ones we employ today have a homology arms that are 1 to 2 kilobases in length that flank the autonomous components of the drive system. But there's some recent work that shows that these can be smaller under special conditions, and that will be important in the further refinement of this technology. So the primary integration is into the chromosome. That is, we built a plasmid that contains all these components, and we actually wanted to get it integrated in vivo into the target chromosomes in the germline. And so microinjections are performed, which introduce this plasmid into a space where it can interact with the germ cells of the organism. And what happens is that we get expression of the guide RNA and the nuclease, they cut, and then we get recombination that mediates integration of this into the chromosome. And so now this is actually in the chromosome, but once again, it carries both the nuclease and the guide RNA, so this can continue in an autonomous fashion. So now we're looking at the chromosomes. Here's one of the homologs, here's the other homolog. It came into one of these chromosomes, but because it carries the information to continue to propagate itself, it will cut this. And basically what we do is we turn a heterozygote into a homozygote. So this idea of outcrosses for testing it, and this idea of going from a heterozygote to a homozygote are sort of the fundamental properties of this gene drive system. So if we look at it, one that we've developed for Anthony Stevens' eye, the system components for autonomous expression involve the Cas9 nuclease driven by a promoter, the vasopromoter, that allows it to go after the cells that are in the germline of the mosquito so that this can be propagated. It also carries the guide RNAs that are consistently expressed, so we can have a germline specific expression of the endonuclease and the guide RNA present, and it targets a gene. So in this particular case, we have a DS red dominant marker that allows us to follow it here. And our particular target here is a gene called kynurenine hydroxylase, or kynurenine monooxygenase, that when mutated and in a homozygous form causes a wide-eye phenotype. So in addition to being able to track the gene drive construct by the dominant DS red marker, we can actually look at faithful integration into the genome based on mutagenesis of the target locus here. And in this particular case, we're actually carrying anti-malaria parasite effector molecules, though we won't be able, unfortunately, to spend much time talking about these today. There's so much information about these that would be way over 50 minutes. But the bottom line here is that this is very similar to what you just saw with our little schematic model here, where we have the chromosome and we have a plasma that allows us to introduce this autonomous system. Going on. So what would the outcomes be? Well, once again, remember, we say it starts as a heterozygote once it's been integrated into the first chromosome. But then the mechanism through homology-directed repair gives us an outcome where we get gene conversion. So we go from 1 copy to 2. However, there's also the possibility that we get something that is called non-homologous injoining, where the Cas9 guide-RNA complex actually will cleave the target here, but there'll be a failure or inability to carry out the homology-directed repair. And oftentimes this will lead to a mutation in the target gene as a consequence of the blunt ends or cut ends of the 2 parts of the gene interacting with one another. Sometimes there's not a faithful repair, and there'll be a mutation here. And these are called non-homologous injoining alleles. The process or the mechanism is non-homologous injoining. So what are the targets for gene drives in males? Well, once again, this occurs in diploid cells. So we're looking in the diploid progenitor cells or gametes. And in the testes here, we have complete mitosis going from the diploid progenitor cell, spermatogonial cell, to the actual actual formation of sperm here on the ends. And so the diploid cells are in this portion of the organ here. This is a close-up here. And so what we're targeting with the drive system is activity in this particular region here. And if we do a male lineage outcross, so these are outcrosses that involve the male introducing it. Once again, we presume it's a heterozygote. We can look and see that actually the Cas9 is where it's supposed to be using the vasopromoter. These are in situ hybridization images by Anita Herman, Gerard Toratis. And you can see that there's a co-localization of the endogenous vasa with the Cas9 expressed by the vasopromoter here. That's the co-localization there. So we're in the right spot for this. And when we do this type of cross, we see that we get very good gene drive. Here the transmission that is from the progeny, 98% of them are DS red, which is what we're looking at here. And the conversion, the amount that goes on is as high as 97%. And these are the phenotypes here. Here's a wild type eye that is from a gene, sorry, here's a wild type eye here, and it's got this very dark color called black or very deep purple. And here's a gene drive animal, and you can see there's a little faint DS red visible here. And it's faint because the eye pigments here make it difficult to actually see the DS red. So we actually score these as larvae where it's easier to see. But that's definitely DS red. So it's very efficient here for this cross. When we ask the same sort of question about female lineages, the thing about the female is that meiosis starts and then is arrested around metaphase 1. And the release from metaphase 1 comes after insemination and oviposition. So there's a long period when these diploid chromosomes are next to one another. And so what we're looking for is expression of the Cas9 guide RNA complexes, both in follicle cells and the developing oocyte where this will occur. And when we look at the in situ images here, we can't see, it's hard to see the Cas9 guide RNA complex, Cas9 guide RNA complexes in the actual oocyte here, mainly because this is in situ hybridization. So this is RNA expression. If we're looking to do some protein localization, but we can see that the RNA is expressed by the vascular promoter in the nurse cells, which provide materials for the developing oocyte. And so we get the type of expression that we're expecting here. So here we're starting with a heterozygous female and crossing it with a wild type male. And once again, we get really good levels of gene drive. Transmission is 98%, the actual conversion rate is 97%. However, interestingly, most of these females actually have a wide-eye phenotype, and that's what we're seeing here. And this is the wide eye, and here now you can see that the DS red is really easy to see because we don't have the wild type pigment occluding our ability to see this. We also see another phenotype, which is called mosaic, where we see patches of apparently wild type cells in the background of dried positive white eye cells. And we see here an obvious mosaic, and then we see ones where we see intermittent amount of expression of the intermittent amount of expression of the wild type gene product, which causes the eye pigment color. And we presume that this is a consequence of large patches of wild type expressing cells somewhere in the body, and the fact that the product of this gene here is not cell autonomous. It can freely diffuse between cells. And we can kind of see that as a very dark cells here and then diffusion into adjacent cells here. All right, so we get the mosaic pattern. These will become significant later. But we see something else here. If we do further crosses from the female lineage, so these are males and females derived from female lineages, we see that there's in subsequent generation coming from these progeny of females that were originally carrying the drive element, we see a diminution of the transmission and conversion frequencies. And so the drive essentially is stalling. And this happens whether it's a male that's bringing it into this female or a female that is expressing it. And so it made us think that something interesting was happening as a consequence of the compartmentalization of the somatic tissues versus the germline tissues. So what we think is happening in the male lineages, that there's germline restricted localization of the Cas9 in the nuclease. So we have a male here crossing with the female's providing the egg. This is the egg here. This is one of her chromosomes here. It's a wild type chromosome. The male's providing the drive system. It comes in, and there's a compartmentalization of the somatic cells and the germline cells, because the germline, the Cas9 expression is germline specific. We have the opportunity for gene drive to occur here, but we don't see it in the somatic cells because there's no Cas9 guide RNA complexes. And when this organism grows up to be an adult, we see dark eyes because there's a wild type allele on the paternal chromosome coming in. It gives us the dark eyes, despite the fact that there's this drive occurring in the germline. And they will look DS red because the somatic cells are carrying one copy of the gene drive system. In a female, it's different because the female, in this particular case, is carrying the drive system, and she puts the Cas9 guide RNA complexes all throughout the egg. And when the male sperm comes in, there's a prolonged period of time where it's not close enough to the female chromosome to use the female chromosome as a template for homology-directed repair. So what this does is favor the production of these non-homologous in-joining mutations. And so when we get somatic tissues and germline tissues later on, similar to what we see here, this is now mutant. And when this develops into an adult, we actually see a wide eye because we have a mutation caused by non-homologous in-joining, and then the integration of the the integrated gene drive construct. In addition, the drive dynamics are damaged because there are resistant alleles here, because this is a damaged allele. The target site may not be sufficient for the guide RNA to recognize, and so the drive decreases. And then we saw that with the reduction in drive in the previous slides. Well, there's other stuff going on. No one said it would be easy. So basically, when we did these initial crosses, the wild type females yielded quite a few progeny. And in similar experiments, there were almost 80% fewer progeny when the progeny were coming from the wide-eyed females. Okay, and we also saw that it was only the wide-eyed females that had mosaic progeny. So that these are the mosaic here that are coming through. Okay, and that'll be useful later on to remember that. So why so few progeny from wide-eyed females? So we looked at the survival, fertility, and fecundity after a blood meal in wild-type heterozygous sorry, this should be heterozygous positive, not minus there, I apologize. And then wide-eyed mosquitoes, and we can see that there's a significant reduction in survivability, number of eggs that they lay, and the average, the number of females that lay eggs, and the average number of eggs that are laid, and then the surviving larvae. So in these two cases here, where we have a wild type and a wild type heterozygote with a wild type allele, we got over 4,000 progeny, and here we only got 30. All right, so there's a sufficient load associated with this combination here, where we have mutations in both the in both copies of the target gene. So it brought up the interesting question about how much of the female has to be this to actually see this phenotype when we start talking about mosaics. All right, so what do we think is happening? Well, the pathway that the KH gene is involved in the metabolism of tryptophan. Tryptophan goes through a number of catalytic steps. This is our target gene here. It's a cinnabar ortholog. It's the ortholog to cinnabar from Drosophila. And basically, when you have this mutation, you prevent the formation of xanthurenic acid. So in work done by Pedro Oliveira's group, they've shown that when proteins are digested, they release individual amino acids, and some of these amino acids actually are toxic: proline, tyrosine, and tryptophan, which happens to be the one we're interested in here. In addition, heme is released because this is a blood meal, and the heme is involved in producing reactive oxygen species. But some of these are still within the midgut. Some of them actually get into the midgut cells, and this can be dangerous because, of course, they're useful for killing bacteria, but they're also highly reactive, can be damaging to the cells. So the production of xanthurenic acid mitigates some of this, because the xanthurenic acid can block the activity of some of the reactive oxygen species. And so we thought that this reduction in female survival, fecundity, and fertility might be a consequence of the absence of xanthurenic acid, and that turns out to be true. And we're looking at the percentage of females surviving after a blood meal, and the wild types here are somewhere around 93%. If they are missing, these are completely wide-eyed mosquitoes, the survival goes way down below 50%. And if we supplement with the wild types with xanthurenic acid, we can actually make them survive a little bit better than the blood meal. But if we supplement the wide-eye ones, we can actually bring them up to near wild type levels. So xanthurenic acid is clearly having an effect here. All right, so we can take these mosquitoes that we've made. This one is called ASMCR KH2. It's also called NRECH, we'll see later on. But this is the one that has the dominant marker for DS red for us to be able to follow it. And we can do cage studies to ask what's the dynamics of gene drive in these mosquitoes. So here's a release where we have one gene drive male for every wild type male, and then 100 females, which they can make. Here's where we have one gene drive male for 3 females and as many 100 females before. And here's a 1:9 ratio, so only 9 gene drive males with 90 wild type males and 100 females. So we set up 3 replicate cages, and we blood feed and we collect eggs. And then the first thing we do is pull out larvae. And we use 200 of those larvae to populate the next generation cages. So these are discrete generations. It's not a continuous generation. It's new cages every time. And then the rest, we look for population size and we screen for phenotypes. So 500 larvae are screened for their DS red and their Carnivore hydroxylase phenotype, white cage phenotype. We look at pupae for screening for sex, male versus females. And the drive efficiency basically is the frequency of DS red versus non-DS red. And then we look for non-homologous end-joining alleles, which would be resistant to the gene drive, which would have no DS red and have a wild type eye, or no DS red and have a white eye. And this is what we see. So here's the 1:1 male cages, here's the 1:3, and here's the 1:9. And you can see that the percentage of DS red following release in all of these goes up very quickly for the 1:1s and 1:3, not so quickly for the 1:9s. And at some point, it approaches full introduction. And when it goes full introduction, that's every mosquito having at least 1 copy. And from there, they go on to become homozygous. And you can see something interesting in at least 2 of these cages in the 1:1s, the populations crash. So this is the number of mosquitoes. And they're up here and then they come down and go to 0. So these 2 go to 0. All right, now this one comes up for a while and then comes down and just sort of staggers along here for a while. The same thing for the one releases. 2 of the 3 cages led to population crashing here. Okay, and for the 1:9s, they got very close, but they didn't go to homozygosity. And then the amount of DS red decreased and the populations here actually recovered. So these are the ones that crashed. These are the ones that got close and then started to recover. And then there's one very interesting one here where it went up and then started to come down. And this is the going up and down here is the percentage of DS red. And then the population size was going down and then came back up, okay, once the numbers of DS reds went down. So we'll get back to this shortly. But the interesting observation is it is possible to drive these to 100% introduction at these frequencies here, and that as a result of that, the populations will crash. So the question then is that during this process, do we actually generate drive-resistant non-homologous end-joining alleles? And the answer is yes. And here's a long table. And I was going to make a joke about going through it, but I won't. Basically, the summary is that we found 50 different wide-eye non-homologous end-joining alleles from 185 mosquitoes. 10 of the 50 originated independently in more than 1 cage. So these are things that are recurrent. We found a number of isoallelic variants in the sense that we had homozygotes that had a single variant, and we found a number of heteroallelic combinations, that is different combinations of these white eye causing alleles. We found 12 distinct in-frame mutations, which was interesting because these are in-frame, but they still gave us white eyes. And it's really difficult to calculate a frequency per generation of these alleles. And we can talk about that in the discussion, why it's so difficult. But we made an estimate, it was about 5% generation and all per generation in all these cages. So these are the white eye ones, no DS red. What about black eyes with no DS red? Well, black eyes with no DS red could just be wild types. But as you get along in the cage trials, the wild types should be gone because the genotype is really efficient. And so we looked at black-eyed mosquitoes in later cage generations, and we found 2 mutations that would give us a black eye. One of them was a synonymous mutation that conserved the tyrosine, and another one was an in-frame substitution that changed a glycine to an alanine. And they were very close to one another. In fact, they were right adjacent to one another. All right. And interestingly enough, if we looked at these two alleles, these are black eye, presumably dry resistant alleles because they show up later on. We can see that in that one cage that we saw back here, where the dry came up and went down and the population went down and then came back up. This is because one particular combination of alleles here, this 83, which is this particular allele, ended up dominating the cage. So in this particular case, a resistant allele halted the gene drive here. Okay. But what happens if we rescue this load that's associated with the white eyes? So what we have here is a circumstance where we wanted to recode the KH gene to restore wild type enzyme activity, and we mutated the guide RNA site in this gene, this recoded region, to be dry resistant. And we use a technology that's been called swap, where you use two guide RNAs to cut at different positions in the target here, and you supply on a plasmid the sequences that you would like to recombine in. And we call it swap to differentiate it from cassette-mediated exchange because the fundamental mechanism is actually different. All right, so the starting animal has a red eye, and we're replacing it by GFP, and we get this what's called Reckh KH, which now has a green eye, a wild type eye because it's carrying a full wild type KH gene here. Okay, and what happens, what do we see? Well, we actually rescue the female reproductive phenotype. So here's the control. We're just looking at eggs per female and larvae per female. These are actually related, obviously, because you get the larvae from eggs. But these are two of the numbers that we have here. But if you look at the control, you can see that there's a certain number of eggs and a certain number of larvae that are produced. If you look at the NREC, which was the one we saw before that does not have the recoded, you can see they were reduced, and we saw data in the table relating to that. But when we put in that recoded KH gene, we actually restore the wild type levels of activity. And when we look then at the drive properties of this, we find that, and once again, we're differentiating between the male and female lineages. When we look in the male lineages, we see that we get almost, well, we get 100% transmission and HDR from both males and females when these animals here are derived from a male lineage. When we look at a female lineage, we see that, like we saw with the other experiment, the non-recoded one, that we eventually see a reduction. And if we can, here's the data from a similar sort of experiment from the non-recoded here. And interesting here, we don't see very many mosaics in the recoded one. And that actually makes sense, right? Because we're providing a wild type copy of the KH gene here. So there's wild type enzyme around. So we shouldn't expect to see a lot of the white-eye phenotype because it's rescued by this particular gene here. But we actually see quite a bit, of course, in the non-recoded ones. All right. Interesting, though, we saw quite a large number of white-eye mosquitoes from female lineages where we expected not to see that because they have a wild type copy here. So what that means is that somehow from this female, these don't have a GFP. Sorry, take that back. They don't have a GFP. So the female likely had a mutation in the chromosome that was to be targeted by the gene drive system. I take it back. These are gene drive GFP minus, so you don't expect a wild type copy to be there. But it was very high. It was 25%. And we compare that to what we saw in this case here, and it was also very high, 40.3%. So this is a lot of generation of non-homologous end-joining alleles, at least in the somatic tissues where we're assaying it for the white-eye phenotype. So when we ask what happens in laboratory cages within the non-recoded, we have the 1-to-1 release, the 1-to-3 release, and 1-to-9 release. And you can see that they all go up to full introduction and homozygosity, even the ones at 9. The population size move up and down, but they stay about the same as the generations go on. So this can actually rescue the rescue system actually works very low when we compare it to what we saw before. So the green lines are the Reckh, and these are the non-recoded. Here are the two that crashed here. Here are the two that crashed here. Here are the ones that kind of petered along. But you can see that these recoded ones work very well, especially compared to the 1 to 9 release. All right, so what are the possible phenotypes and genotypes? Well, there's a whole bunch of combinations, and there's a lot of information on this slide, but the thing to look at is the green. And the reason the greens are different shades is because these are the different cages. But we have a 1 to 1, 1 to 3, 1 to 10, and what you can see is the green starts somewhere and end up being most of what the phenotypes that we see. And when we look at the genotypes, this is GFP positive, which is all the green, and black eyes, which would be a wild type allele. And there's a number of ways that this can occur. There's some GFP positives that have white eyes, very rare, only 20 out of 79,000. And there's some GFP positives that were mosaic. These are even more rare, 1 out of 79,000 that we found. All right. These are the non-GFPs. So they start out being wild type, which you can see all the wild types here in these corners. But as the generations go along, of course, they are targeted by the gene drive and are displaced here. We saw about 2% of the GFP minuses were white, and they show up here along the top. Okay. Interestingly enough, they increase in frequency for a little while, and they start to go away. They increase and then start to go away. Increase and start to go away. That's a general feature throughout, and we'll get to that right away here. But we also saw mosaics that came from this. So the question is, what's going on with these whites? I mean, they're resistant to drive. One might expect that there might be circumstances, especially on this side, where they might actually be able to increase in frequency in the population. But we've invoked a phenomenon called lethal sterile mosaicism. This was first discovered by Ethan Beer working in Drosophila, where if you have maternal deposition of the Cas-Guinar complexes in the fertilized egg, they accumulate, as we talked about before, in the eggs under the expression of vasa here. So we can see this. So females that are heterozygous can carry a copy of the gene drive locus. And if we, this is the incoming locus from the male here, so it's wild type. The blue is the maternally derived Cas9-Guinar complexes. And this white here is a non-homologous injoining induced non-functional resistant allele. So what happens is that this female is positive for green. She may or may not have her second allele be either a gene drive allele or injoining mutation here, but we depict it as an injoining mutation. And so when this comes into an egg and the sperm come in and the male chromosome comes in, there's two possibilities. One is this allele can remain wild type, or two, it can be mutagenized to a non-homologous injoining allele. It's probably not identical to the one the female provided, but becomes a heteroallelic combination. And this can happen throughout the egg, and as a consequence, you can get a mosaic, and this is the mosaic pattern here that we see as a consequence of that. And there's no propagation of these alleles through drive, but they're maintained if there's sufficient numbers of wild types. However, if there's insufficient number of wild types, then it's possible that this would lead this subsequent female to have reduced survivability following a blood meal. So somatic cells of progeny with somatic cells of progeny with drives that are mosaic. So we get the same thing from a lineage here where we get the drive coming in, and the drive then can do one of three things. It can either drive to turn this to a drive locus. It can either mutate it to non-homologous end joining, or it can leave it intact. And that's what we see here. They'll have green eyes as a consequence of that. Okay, but because it's been rescued, they'll have dark eyes. Okay, so in this particular case here, all the phenotypes are GFP plus, and the females survive and are fit for reproduction. In this particular case here, the phenotypes are white eye or mosaic, and females may have reduced survival following a blood meal. And the diploid cells that end up in the germline then can impact the transmission dynamics. So does this drive generate potential drive-resistant alleles? So the drive-resistant ones will have to be black-eyed, okay? Because we've just shown that with these data here, that we lose the whites with time, okay? So the whites are probably eliminated through this lethal sterile mosaicism. So the ones that persist, the ones that are going to be resistant ones, have to have black eyes. Okay, so we looked at black-eyed ones in late cages, G16, and we found that they were all homozygotes with a glutamine to alanine substitution that affects the PAM site. And if they affect the PAM site, we expect these to be drive resistant. And the vast majority, well, not the vast majority, but about half of these were homozygotes for this Q330A. The heterozygotes are mostly Q330A, but they also have other types of them, but they all affect the PAM site. So these are highly likely to be drive-resistant alleles. So here's our target target site in the wild-type chromosomes here, and this is the amino acid sequence here, the nucleic acid sequence. And the question is, can these drive-resistant alleles outcompete can the drive-resistant alleles essentially outcompete the drive system? Here's our guide RNA, and here's the proposed cleavage site. And here's that Q330A substitution here, and you can see where it impacts the PAM site here. And the phenotype is a black eye here, okay? So what we did is we made heterozygotes of the drive system with this particular resistant allele. So we have a heterozygote that carries a gene drive package, and then on the other chromosome it has a resistant allele. And we basically did an intercross. And this is the Punnett square for those of you who enjoy this sort of thing. And you can see that when we do a cross of a female that's heterozygote with a male that's heterozygote here, we expect a 3-to-1 ratio of green to wild type, okay? And then we would just take this and then run it for 6 different generations and ask what happens as a consequence of that. So they all start out being green at 0. And after a single generation, we have a frequency here of the resistance homozygote and then the remaining drive competent animals. And you can see that this is the ideal 3-to-1 ratio here in the dotted lines. And you can see that with time, actually the drive outcompetes this wild type resistant allele. So why is that? It suggests that perhaps there's a load despite the fact that it looks wild type because it is black eye, that there's a load potentially associated with this. And we think that's because looking at a number of mutations that we found in this region. So here's amino acid 328, here's amino acid 330, there's mutations in 329, combinations of 329, 328. Some of them are black eye, which is the putative wild type phenotype, and some of them are white eye, which is a mutant phenotype. All right, so we think that basically we're targeting an important region in the protein that is made by chinerin hydroxylase, and as a consequence of that, even though it's makes enough to give a black eye, it may not be sufficient to confer a number of pleiotropic effects that would affect its fitness. So with time, we would expect to lose that. So that's a possible model for that. Okay. Well, can we change the target gene? So we've done a lot of work with the KMO here. And issues associated with the fitness of mutations in this particular gene. We decided we would try to bypass that by looking at a gene that's downstream. And we switched over to Anopheles gamete for that. Now the target here is the heme peroxidase gene, which is lower in the chain here and allows the production of xanthorenic acid. It too has an eye color phenotype, we'll see in a second as well, where the homozygote is this sort of reddish looking color and the wild type looks like that. So a very similar situation. We have a plasmid that allows us to introduce it, our target gene. In this particular case, it has cyanfluorescent protein, and we can score that in larvae. You can see a very nice image here with that. And then the eye color phenotype is this sort of pale color, and we can see that in pupae. And then we see it in newly emerged adults, but as the adults grow older, the eye darkens. And so there obviously is some alternate pathway supplementing what's needed for this eye pigmentation. But there's clear phenotypes in the subadult stages. This is driven by the nanos promoter. And when we look at Cas9 expression driven by the nanos promoter, here's the endogenous nanos messenger RNA, and we can see we get really significant overlap. So we're in the region where we want for both the testes and in the developing oocytes to get this in the diploid cells. When we do laboratory cage trials, 1-to-1 releases, 1-to-3, and 1-to-9 releases, we can go to 100% in all of these, okay? And we've looked at the fitness aspects of these particular integration. This particular integration events, and we find really no fitness effects that certainly impair the ability of the drive locus to actually work. So one of the things we were able to do with this that we weren't able to do with the previous two trials is that there are a lot of existing Anopheles gamete strains, and we were able to look at natural target site variants. And here is the sequence of our target site guide RNA listed from the PAM site all the way through the additional nucleotides here. And we were able, this is collaboration with the group at Davis led by Greg Lanzaro, we were able to look for variants in the PAM target site in wild type populations of Anopheles gamete and also closely related species, Anopheles colucci. And these are the variants here. They're generally low, okay, but we found 5 of them that were sufficiently high enough to follow up. We looked at this one, even though it's not very high, because it's actually in the PAM site. And the rest of these are pretty high here. And what we did is we made synthetic target sites that contain variants of the PAM, variants of the target site, and cloned them and cloned them into a plasmid. And that plasmid has an NCO1 site, and what you can do is linearize the plasmid based on the NCO1, so that linearizes it, and then you can ask, do you get cleavage then at the target site? So for our variants that we saw, we have 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. And interestingly enough, they were all cleavable by in vitro by Cas9 guide RNA complexes. Now, number 1 here, which is in the PAM site, wasn't cleaved qualitatively as well as the rest. Okay, but we don't know what this means in terms of implications for gene drive. And what we're doing now is we're getting strains of mosquitoes that have a high frequency of these variants in them, and we're going to be doing the drive experiments to ask, can we drive into these variant containing populations? And the expectation is that it will drive, but with slightly slower dynamics than what we saw with the canonical target that is the control target here. We also look for off-target sites. So these are regions in the genome that look similar to the target site, but vary. Here's a control here which lacks the PAM site, and these are 5 different variants that existed in a population that we have here. And we asked, do we see cleavage at these sites? And for only one, which is off-target 1, did we actually see cleavage. So the off-target sites, going to someplace other than the target site here doesn't seem to be a very high possibility or probability. All right, so finishing off here, this leads us to this concept of how good does this actually have to be useful? Well, it has to meet safety and efficacy criteria. It has to have elements that could address regulatory and social criteria. And for that, we developed target product profiles, TPPs. At this point, point out it's no longer an academic exercise. And what we are trying to do is identify ideal characteristics of what the dry system would be like, and then ask what's minimally acceptable in terms, or essential for use. And so we've done that. We don't have time to go through the tables, they're quite long, but that last system I showed you and the recoded system meet a lot of the requirements in a target product profile for moving forward. And a lot of this is informed by modeling when we can't actually do the experiments. So what are some of the take-home messages today? Well, we think that these small cage trial formats are useful for testing some of the gene drive systems. I mean, it's obvious they do not mimic nature, okay? They aren't designed to do that. But we can learn a lot about the systems from the types of experiments that we've defined. All the systems we developed are very efficient in mosquitoes, both in Stevens eye and Gamby. And we target both an essential gene, which is the KH, and the non-essential gene, which is the gene in Adoptis gamby. And the rescue system worked very well for us. And we like to think perhaps that the Cas9 guide RNA mediated resistant target sites may not be the problem that many people think they are. They seem to be generated at high frequencies and they get into somatic tissues, but when you actually do a cage trial, except for in the non-recoded circumstance, they were not able to take over the cages in 9 replicates for 3 different experiments. 8 replicants of 3 different experiments. The one experiment, they were able to do that. So this is the last slide. It's to remind you that we don't think that our technologies alone are going to be successful for eradicating malaria. It's going to require a combination of all of these to get to this particular goal here. And so we're not trying to advance the idea of population modification as being the sole thing that can be done in order to achieve eradication. So I'll stop there, I think almost on time, and thank you, and we'll open it up for questions. I will stop share right now. Actually, you know what, I'll leave share up because maybe people will have questions. I'll just end the show. And we'll turn it back over to David. Uh-oh. I think I have to stop share to, oh man. Got it, David? Was it coming through? Did it drop off and I never heard? Hello?
Okay, sorry.
Oh, okay.
There we go. Yep.
David,
Sorry.
you're killing me, man. For a moment, I thought maybe it failed at the very first minute.
Yeah, sorry. Well, you know, two mute buttons got me screwed up. So,
right.
So let me just encourage the listeners to type in questions for Tony. Tony, I can start with one, maybe just a general question or a broad question as to the general strategy for picking pigment pathway genes in as targets. I know you've picked, you know, the cinnabar and now the cardinal.
Right. So the original idea was to have a marker that would allow us to, in a phenotypic way, monitor fidelity of integration, so that if it was going to the right site and it became homozygous, we would get a phenotype that we could score. And so that made it easy to sort through all the possible dominant marker plus, so everyone's carrying a dominant marker, sort through all those and find the ones that went to exactly where we wanted to go. Now, down the road, we've also argued that, especially for cardinal, that it might be something that could be useful in the field for tracking transgenic mosquitoes using only light microscopy and actually probably just visually looking at them. I think people can get adept at seeing the light color phenotype in just a tray without a microscope. But the idea initially was to be able to monitor that group of insects that were carrying the gene drive that went to homozygosity. So if they were heterozygous, they would still have a wild type eye, and if they went to, you know, they could be GFP plus heterozygotes, still have a wild type eye, but when they went to homozygous, the eye color would change.
So in the field conditions, you don't expect that to have necessarily a fitness cost under field conditions?
We anticipate that it won't, but even if it did, the drive dynamics is strong enough, we think, that it can overcome the major fitness issues, as we saw with the Reckh. And so way back early on, when we were talking about you know, rules of the road of gene drive, we talked about the fact that they could actually tolerate quite a bit of fitness as long as the drive was strong enough. And there were, at least for transposable element-based gene drives, the modeling suggested that a bias of just a few percent would be enough to bring would be enough for the drive locus to succeed. And we have, you know, drive efficiencies that are well above that. So for population modification, we don't think it's going to be a problem. It's a different story for population suppression.
Right. And so one question relates to the resistant alleles not increasing in frequency in your population. Is that a function of the fact that you're doing a modification and not suppression?
So it's a combination of two things. I think the modification and strategy gives us a leg up there, and then at least for targeting the kynurenine hydroxylase, where resistant alleles may cause a disruption of the gene function, and if they become homozygous, we lose them. So this sort of lethal sterile mosaicism are two things that we think make that work. Okay, so once again, the modification strategy, I think, has a slight leg up on, well, not a slight, a big leg up on the issues associated with the, you know, with the generation of resistant alleles. I think I answered the question, maybe not.
Yep. Well, I'm not seeing a lot of questions from our audience, Tony. So
I want to hear from Ryan. Let's see, it just popped up. Okay. So Ryan's, oh, I'm sorry, I'm usurping your job, David. I apologize.
Yeah, go ahead. Go ahead. Whatever you got. Mm-hmm.
Yeah, so good to see you, Ryan. He's asking about whether or not the KH genes we mentioned were ever challenged with Plasmodium. And because their disruption of the XA pathway also can impair gamete activation and subsequent parasite development. So there's a series of experiments that were done by people who showed that xanthorrhinic acid can contribute significantly to gamete activation in experimental systems of malaria parasites. And so We didn't find that in work that we did. Okay, we had a strain of mosquitoes that was the white-eye strain of Bala, which was deficient in xanthine, which was deficient in the enzyme and didn't produce very many, didn't produce very much XA at all. However, it could be infected with Plasmodium gallinaceum parasites. Okay, so we were a little bit, puzzled by those results. Okay, having said that, there are two additional sets of experiments that show in Stephen's eye and I think in Gamby that the absence of XA does impair significantly parasite development. And if that's the case, then we get a little extra then in using that as a target locus. So we're testing now whether or not these insects show additional resistance to malaria parasites, but that work is ongoing. I get shorter answers here, I'm sorry.
That's okay. Okay, now I'm not seeing any other questions, and I'm not sure that I'm
so Suresh
see the questions here.
says,
Yeah,
looks
okay.
like
So
the gene drive mosquitoes
I'm not seeing those. So go ahead and Tony and take those as they come in.
They're coming up on me. It says it looks like the gene drive mosquitoes are as good as wild type in mating. Is this correct? So we actually found no significant mating differences between wild type and the transgenic mosquitoes. Now, when you look at a number of different parameters, sometimes the wild types score a little bit better than the transgenics, and sometimes the transgenics score a little bit better than the wild types. And so I'm reluctant to make a categorical statement that the transgenics are better than wild types or vice versa, wild types are better transgenics. It's just the net overall effect is one where they seem to be at least equally competitive. And if you're going to hedge it a little bit, the transgenics might be just a little bit more competitive in some of the experiments we did. But we kind of summarize all that heterogeneity as saying there doesn't seem to be a significant impact of the transgenes on competitiveness in the Reckh, sorry, in the recoded one and in the Gamble experiments. Obviously, with that first one where the females were experiencing significant reductions in fertility and fecundity and survival after blood meal, there was a big impact. Okay, only in the females, not in the males. But the expression here are as good as wild type in mating seems to be true. Yes. Other questions or comments?
Let's see here. Okay, I guess there's one more question, which I'm not seeing, but can you see it, Tony?
I can't actually, hang on. I see one from Ryan, one from Suresh. Can't.
Okay, so maybe not.
Yeah. All right.
Okay.
Well, once
Right.
again, thank everybody.
Yeah. Thank you, Tony, for doing this for sure. And if anybody has any other questions for Tony, you can certainly email. You can email the GeneConvene, and we're happy to pass them on to him, and we can post those questions with the recorded video.
Yeah. So just as a last comment, there's obviously now a lot of interest in what the effector molecules are doing, and so the next phase of the work, we think the drive system is pretty much nailed down at this point. There are risk assessment, regulatory, community engagement aspects of it that are continuing to be addressed, but the big in-lab challenge now is to pair these up with effector molecules that will show really good efficacy in getting rid of the malaria parasites. And accompanying this are some really interesting thoughts about just how good the effector molecules have to be in terms of knocking down parasites. We've always thought that we had to get to, at least I've always thought, that we've had to get the parasites down to 0 in the salivary glands. But there are a lot of experiments and data that are accumulating and being analyzed, which suggests that there may be threshold levels for parasites in salivary glands, and so that it doesn't have to go to 0. And if that's the case, then for some of the molecules we have that we can talk about in a future date, we're very close to having them.
While you were talking, we had one question came in from Derek, and so he says, "Great talk, Tony." So on competitiveness of transgenics, of course, this was against the lab strains.
Yes.
With the Oxitec transgenic releases, there was a significant difference in fitness to field strains, not seen in lab results.
Yes, very good. Thanks for bringing it up, Derek. So we're actively doing those experiments now. We've obtained 2 types of for the Gambi work, we've obtained 2 types of additional strains. One are colonized field variants from different locations. So they're colonized, but their provenance is quite different. And so we're asking the question of how effectively the drive works in these other strains of Anopheles gambi, and then also colucci, a closely related member of the species complex that produces fertile hybrids. And then we're also asking, what do we see if we look at other closely related mosquitoes for which there is the Haldane bias, where the males are impaired as hybrids, but the females are okay. And so we're looking at those in terms of introgression and asking to move into that. In terms of getting recently colonized field species, we have collaborations in a couple of prospective field sites that we're looking at, and the field component of the team, which unfortunately are poorly represented today, is attempting to get access to those mosquitoes and colonize them. We'd obviously be a lot further along, except for, of course, that we can't travel now. And so as soon as the traveling conditions loosen up, we'll be trying those experiments. But that's a really good point, Derek, and it's part of the target product profile to show that these are equally efficient in recently derived wild populations. Really long answer, sorry, man.
Yep. Okay. I'm not seeing any questions now for sure. So maybe we can just wrap this up. Let me say thank you very much, Tony, for doing this. Let me say to the audience that our next webinar in this series will be a week from today at the same time. Our speaker will be Dr. Bruce Hay from Caltech University. So with that, Tony, I'm going to say thank you very much, and we'll close this session of the webinar.
Okay, great. Thanks a lot.
Thanks.